Bismillahirrahmanirrahim. This is Hafiz Muzammil Rahman, and this is our lecture regarding the drug designing. And this is the first lecture in which we will talk about the protein ligand docking. That is the step of the drug designing and discovery, and where it lies, and how we will do this, and what is basically the protein ligand docking. First of all, we will go a little bit with the concepts. What is the protein ligand docking? Why we need the docking process, and so on. So we will discuss a little bit that, and then we will go to the practical part of today's tutorial as well. So first of all, I'll introduce you to the protein ligand docking. Basically, this is computer aided drug design. We have different option in the CAD or computer aided drug designing. If we know the ligand, then and we know the structure of the protein as well, then we can go for the structure based drug design. And if we don't know the ligand, so we will go for the de novo design. And if we know the ligand and don't know the structure of the protein, then we can go for the ligand based drug designing, and so on. We will focus in today's lecture, that is the protein ligand docking. And in the coming tutorials or lecture, we will discuss all these things that will cover the whole computer aided drug designing. So we are moving towards the protein ligand docking. What is protein ligand docking? Is basically the part of the CAD, and that is under the umbrella of structure based drug design method. What is the meaning of structure here? From the structure, we mean the protein structure which we use as a target. And then in the computational method, we use that mimic the binding of a ligand to the protein. What is the ligand? Ligand can be a small molecule, it can be a peptide, it can be any inhibitor. So we have given the protein. We can have two cases. If we have a protein, so we know the binding side of that protein. What we will do, we will try to interact both of these, the ligand and the protein, at the binding site. Then we will check the interaction of the ligand and the protein at the binding site, and that is called the pose. We will predict the pose of a molecule in the binding site, how the ligand is aligned into the binding site. That is called the pose. Like this molecule is aligned in this way, in a bending manner. So this is the pose of the ligand into the binding site. And the other thing which we will predict after the protein ligand docking, that is the binding affinity or the score, that representing the strength of the binding. In a simple way, we can say that in the protein ligand docking, we will try to find the interaction between the ligand and the protein at the binding site, or you can say it the active site of the protein. And then we will find how the ligand is lying in the binding site, and then we will find how much these interactions are formed, like whether there are hydrophobic interaction, there are hydrogen bonding and other. And then in the last, we will find the binding affinity, the strength of these binding interaction into the ligand, into the protein binding site. So these will be discussed in today's tutorial. So binding site means the active site, the part of the protein where the ligand bind. Generally, a cavity on a protein surface, that is used at the binding site. Or if we have a protein structure and we have modeled the protein structure by ourselves, like I have told you in my previous tutorials how to modeled your protein. If you have a sequence of a protein, in today's time we can predict the structure of the protein computationally as well. And if you have predicted the structure computationally by the homology modeling or by the AlphaFold or by the Rosetta fold, etc., you can go to my other tutorials about the structure prediction as well. And if you have predicted the structure and you don't know the binding site, so there are other ways. There are some server, you can go to the iTester, and there are some server which help you to find out the binding pocket as well. I will share the link of these sites from where you can find out the binding pocket of your protein in the description of this video. So that can be identified by looking at the crystal structure of the protein bound with a non-inhibitor. If you got a protein structure from the PDB and the ligand is already bound to it, so where the ligand is bounded or already bound, that will be the binding site of your protein. Pose mean, as I have already discussed, that is the binding mode, the geometry of the ligand in the binding site. So what do you mean by geometry? That is the location, that is the orientation, as well as the conformation. You know, the small molecule, they may have some rotation around the single bond. The molecule can be rotated. So after rotation, it can change its orientation and conformation as well. So protein ligand docking is not about the identifying the binding site. It is about the to find out the interaction between the ligand and the protein or target site. So this is a picture of the docking, in which this is our ligand, and it is bind in that cavity and in that pocket, and it make interaction with these residues of the protein, that is arginine 74, arginine 74, and so on, tyrosine, etc. So it can make the interaction with all the residue which are present in the cavity or binding site of the protein. So uses of docking is, the main use of the protein ligand docking are for virtual screening, that is to identify the potential lead compound from a large data set. If you have a large data set of a molecule and you can find out, you want to find out the good inhibitors against that protein, so you can go for the virtual screening. In today's lecture we will just dock one compound with our target protein, but we can also dock with the multiple ligand with our protein. So we can predict a pose prediction. Pose prediction mean if we know the exactly where and how a non-ligand binds, we can see which part are important for binding. Like in this figure, you can see the tyrosine. Sorry, the tyrosine is making interaction with this part, and the arginine is also making interaction, and this amino acid is also making interaction in this whole cavity. So we can see which part are important for binding, and we can suggest the changes to improve the affinity as well. Like we can modify the ligand as well. That will be discussed in the coming tutorial as well, how we can modify a ligand, how we can make the derivatives, we can change the groups, we can add some function group at this side, we can add some other groups in this side to make it possible to find more interaction within the cavity. In a simple word, we have a ligand and we can make the changes in that ligand to find out the interaction with the protein or with the receptor. So we can suggest changes to improve the affinity. So avoid changes that will clash the protein. If we change something like here, and it clashes with the residue of the protein, so we will avoid this change. We will just make the changes that part of the ligand will not clash with the part of the protein. So in today's lecture, in today's practical tutorial, we will find how these can be performed. We are not going to the virtual screening. So the next thing, what is the component of a docking software? As I have already told you, docking is a process to find out the interaction between the ligand and a target protein or receptor. So typically a protein ligand docking software, we do these things in a software. So software consists of two main components which work together to get the results. The first is the search algorithm that generates a large number of poses of a molecule in the binding site. Like if you have a small molecule and you are going to dock that molecule with the protein, so the search algorithm will generate a number of poses of that specific molecule within a cavity or within a binding site. Then you will decide which pose is good, which pose has good interaction. So the next thing is how you will decide which pose is good or not good. That will be decided on the scoring function. You will calculate a score or binding affinity for a particular pose to give what we will find out and what we will get after doing the docking, the pose of the molecule in the binding site, and then we will get the binding affinity or a score representing the strength of the binding of the molecule. I mean, after the docking, we will get a complex of the protein and the ligand. That ligand will be binding in to the cavity of the protein or the binding site of the receptor, and then the software will also give us a binding score or affinity score as well. So these are the important things which we will discuss. So practical aspects, before going to dock our ligand to a protein, it is important to prepare the protein structure. What we mean by protein preparation, like if you have a PDB structure and you have downloaded it from some websites, so it may have something which are not important while you are docking your ligand with the protein. Like it may contain the water molecules, because if the molecule is submitted on the basis of the X-ray crystallography or the electron density, so it may have some other atom, some other ion, some metal ion, some water molecules, some head atoms. So you will remove all these water molecule except those which are into the binding site or which are important for binding as well. I'll discuss it when we will dock it, but you should make your mind, we will remove all the molecule except those which are important for binding of a ligand to the protein. What I mean by this statement, in a simple way we can say that if the molecule if the water molecule is nearer to the binding site or inside the binding site, and our ligand is firstly make linkage with the water molecule, and then the water molecule is further linked to any residue, so that is called the water bridge. In a simple way you can say that our ligand is linked through the water molecule to the receptor or to the residue of the binding site, so that water molecule will be important. And how you will know about that water molecule, you will find these things in our literature as well. So you will remove all the water molecule except where it is known that they play important role in the coordinating to the ligand. So PDB is sometime the PDB structure which you download from any website or server that may have some missing hydrogen. So you will have to add the hydrogen. So many docking program require the protein to have explicit hydrogen. In general, these can be added unambiguously except in the case of acidic or basic chains. Maybe there are some incorrect protonation states, so you will have to change it accordingly. These are yes, the next thing will be you will have to prepare your protein structure as well as the ligand structure. A reasonable 3D ligand structure is required as a starting point. So during docking, the bond length and the angle in the ligand are held fixed, only the torsion angle are changed. The protonation state and the tautomeric form of a particular ligand could influence its hydrogen bonding ability. Like uh, if you have a tautomer of the ligand and it make hydrogen bond and the simple form doesn't make, so that will be very important to know whether in which state you have the ligand and in which state you are using the ligand to dock with the receptor. Either protonate as expected for physiological pH and use as a single tautomer, or generate a dock all possible protonation states and tautomers and return to one with the highest score. There are many software which do that. One is the glide, that is by the Schrodinger. So it formed the 32 conformers or protonation state if you give them a ligand. That is the default in that software. So these things are very important. If you feel it is difficult to understand, so just think about the single bonds. Like here, that single bond is attached with the OH and this OH is removed, so that will be form the ketone from the enol, or this molecule can rotate again around this single bond. So these conformation can change, and there are many things to do like that. So these are both two things, they are very important. You will have to prepare the protein. By the protein preparation we mean you will have to remove all the water molecule and you will have to add the hydrogen. You will correct the missing chain as well. If sometime it happens when you download the structure from the PDB, there are some missing chain as well, so you will have to add these missing residue before going to dock your protein and the ligand. So these are the different conformation of a ligand. So it is important because when we are talking about the orientation or conformation, so like I was talking about the rotation. So it while the part of the ligand rotates, is it change the conformation of the ligand. So the conformation is very important because we have discussed the pose, and the conformation will dependent on the pose, or the conformation will tell you in which dimension or geometry it make bonds, or it make interaction with the receptor cavity. So it is like that. Searching algorithm, we will discuss these things in detail when we will uh, perform these things in a very advanced level. So in today's lecture we will just discuss the basics of the protein ligand structure, and then we will perform the practical part. So now we are going to the practical part. uh, I just suppose that after this whole discussion, you will have an image in your mind about the protein ligand docking. So now we are going to perform all these things which I have discussed. First of all, some software you must have before going to dock, going to docking practical. First of all, you will have to download the MGL tools. How you will get it? You will just type MGL tools on the Google and then click on this, and you will have to download it from this website. If you have a window system, then you can download this one. If you have a Linux, you can download accordingly. If you have a MacBook, you can download accordingly, according to your system. So I have a MacBook, so I have downloaded this version. So next thing is you will go to the Autodock. What how you will be able to go to the Autodock website. Just type Autodock on Google, Autodock download, and it will be like this. Here you can go to the download, and then you can download accordingly, if what system you have. Okay, so this is the proceed to download page. You will have to register yourself first here. This is a registration form. You will have to register it, and then you will go over here, and then you can download it accordingly. If you have a Linux, you can download one of these, and if you have a MacBook, you can click here. If you have a window, you will click over here, and so on. And the next thing is OpenBabel. You will go to the Google and type OpenBabel GUI, download. So from here you can download it. This is the OpenBabel. You can download it from any website. There are many websites which have the OpenBabel, so you can download from anywhere. So you can download it, like when you click download, so it will be downloading, and then you will have to install it. So when you will download MGL tools, Autodock, and the OpenBabel, so you have all the things which are necessary for protein-ligand docking. This is the MGL tool. I have installed it. So this icon will be shown in your system. You will click on this icon, and then this GUI interface will open. Graphic user interface will be open in front of you. Okay, before going to start, you must have a protein. Like I have made a folder by the name docking_tutorial. I have a protein which I have already downloaded, or I have already predicted by the homology modeling or by the AlphaFold. So this is my protein, alpha_gluco.pdb, and this is my ligand. I have already a ligand. You can find out the ligand and the protein on PDB or any other website. You can download the ligand from the Zinc. You can download the ligand from the PubChem. You can download the link from anywhere, or you can download the protein from all these website as well. Let's suppose if you go to the PDB website and you type 6UL7, this is the structure of human kinase in complex with fructose and blah 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 blah. So you can download this protein structure over here, and these are the ligands. You can download this ligand from here, and then you can perform the docking. This is an example. I'm not going to work on this, but this is an example from where you will get the protein structure. When you will click over here, you can click the PDB format, so the PDB structure will be downloaded, and same with this ligand as well. So after downloading the protein in the PDB format or in the PDB-QT format, you will download the ligand as well. Another important thing is, when you will download Autodock, Autodock will contain these two exe file. First one is the Autodock grid 4 and the Autodock 4. These two files will be available when you will download this Autodock tool. When you will download this Autodock, so the folder that will be downloaded, it will contain these two files. I'm just going to zoom this. Okay, these two files, the Autodock 4 and Autogrid, they are very important. So you will copy these two files and paste into the working directory. By the working directory, I mean in which folder you have put your protein structure, your ligand structure, and then these two files, you will have to put also these two files on that specific folder. I am just discussing the very basic things. First, you have downloaded your protein structure from the PDB, and then you have downloaded the ligand. Even you can sketch by yourself your ligand. That is another topic I can show you here. If you have a Mestro or any other software, so you can this is a Mestro which I have already installed, and this is the academic version. So you can sketch it by yourself, like this. This is like, this is a structure. So you can make any structure like this. I have made it. This is the ligand. This is my ligand. So you can make your own ligand as well, or you can download it. And how I have made it, and what is this Mestro, these will be discussed in some other tutorial. In today's lecture, we will focus on the docking, but by just the downloading your protein structure and the ligand structure. If you have downloaded, put it into the folder, give any name to that folder, and put these two files, Autodock 4 and Autogrid 4, in that specific folder. So in today's tutorial, the folder name is docking _ tutorial. This is our folder. In this folder we will do all the analysis. Okay, so now we are moving towards the practical part. Okay, where is the tool? There are many things which are open in my MacBook. This is the GUI interface of the Autodock. So first you will click on the file and then come to the preferences and then click the set. So this is the startup directory. In this place, if you have a window, when you will open any folder like this, I have opened this. So when you will open any folder, in the top of that folder there will be, when you will click over here, there will be a path that will be start from the C slash documents, the name of the folder, etc, etc, etc. When if you are working in the Windows, and if you are working in the Mac, so that will be start from the user slash temp local, etc, etc, etc. So my working directory is user, then the name of my PC is Muzammil, and then I then the desktop, and in the desktop, that the folder is the docking tutorial. So this is the folder. So I have give the path to that directory where my folder is present. Path mean where your folder is present. From the base, from the very starting folder or very starting document, you will lead that software to that folder in which your protein and ligand are present. Like in my case, the first thing is the user, and then the second folder is Muzammil, and in Muzammil there are desktop, and in desktop there is the folder by name docking tutorial. My all data are present in this, and I'll work in this folder as well. So by copying your path, you will paste here, and then you will click on the set. So path is set. Okay. Then you will click on the file and then read a molecule. By reading the molecule, you will click the PDB structure or PDB-QT structure which you have downloaded from the PDB. This is very important. The Autodock work on PDB-QT format. It does not, if you have a ligand in or protein in PDB, so you will have to convert first these into the PDB-QT format. How we will convert it into the PDB-QT format, this is important, and I'll just tell you how you will be able to do this. This alpha underscore gluco.pdb, that is my protein. You suppose you have downloaded any protein structure from the PDB, so that will be into the in the format of the PDB. So I will select that structure, and it is into the PDB format. So I will show you how you will be able to convert this into the PDB-QT. So just open it. What you have done, you have click on the file and then the read molecule, and by the after that you have selected your protein. Okay, so now this is our protein. This is our protein or our receptor. After clicking on this structure, it will be shown in this interface, and now you will prepare this structure. The next step is to prepare the protein structure, which I have discussed already. The protein preparation is very important before the docking. So what you will do in the protein preparation? Yes, just recall what I have told you to do. What you will have to delete the water molecule. So just go to the edit, and here delete water. So you will click on this delete water molecule. So if the water molecule will be present in that protein structure, that will be deleted. In this case, there is no water molecule present in my structure. Okay, so you can also see whether this protein structure contain the water molecule or other atom or het atom that is present along with the PDB structure or not by clicking over here. You can just click here. This is the chain X. In your case it may be A, B, C or whatsoever. So it will show you all the residue of that chain or that protein. Scroll down to the end. When it will end here, if there are some water molecule or het atom present in your protein, so it will be here after these residues. So you can manually select that het atom or water molecule by just clicking this box. Like I have selected this lysine 584. So you can see this. It is represented here. That if I select the upper box, that is 583, so it will this residue will be yellowish. You can see this like this. I have just selected these two residues. If I deselect, that will be disappear, that will be disappear. You can select any residue into that PDB structure. So we will go to the edit and delete water molecule. What we are doing, student, what we are doing, we are preparing the structure, the protein structure for docking. So what we have done so far, we have just shown that protein structure into this interface by clicking the file, then the read molecule, and selected that structure. And the first step is go to the edit. I'm repeating it again and again. Go to the edit, delete water molecule. Okay. The next step is go to the MISC, and then click it. Here there is an option check for missing atoms. We are going to check whether this PDB structure has some missing atom or not. So we will click on it. If there are some missing residues or atom, it will be shown over here in this space. But in my case, there is no missing residues, zero missing residues. Let's suppose if there are some missing residues, it will be appear over here, and then you will click on this, select all residues, which will appear here by clicking this. So I have clicked, but it shows no residue, missing atoms. Okay, so you will have to click this and then dismiss this. After this, again go to the MISC and click on the repair missing atom. In the first case, you have checked whether this structure has some missing atom or not, and then if you find some missing atom, and you will have an idea about the residue as well. You will know the residue name by clicking the check for missing atom, and the next step you will repair these missing atoms. Then you will click over here and it will automatically repair all these missing residues. So in my case there is no missing residues, so there is no repair. Okay, after this again go to the edit and go to the hydrogen, edit and then hydrogen. Click on this and then the first option is add. Click on this add and then click to the polar. So you will add the polar hydrogen because they are important for the interaction when you will dock your protein structure with the ligand. So click the polar and click on OK. Just look at this surface. What is going to happen after clicking over there? I have clicked. Yes, these residues, these there was some change. These hydrogen, polar hydrogen, are added in this structure now. If you want to check it again, go to the hydrogen, add, and then polar only, and click. So now there is no hydrogen to add because we have already added the polar hydrogen into this residue or into this protein structure. After this, we will go to we have deleted the water molecule, we have checked the missing atoms, and now we have added the polar hydrogens. After this, we will check the charge on the whole protein structure. So go to the charge and we will add the Coleman. You can add the Coleman charges. Some scientists says that, to add compute gesture if you have a small ligand, etc. But I would recommend to add the Coleman charges. So it is showing -2 total Coleman charge added, that is equal to -2. So click OK. So charge has been added. Again, you want to check whether this charge is added to the whole protein or there are some residue which are left. So go to the charge again, and this time there is an option, check total on residue. So you are going to check the total charge on residue. You are going to check whether some residues are left by adding the charges or not. So there is no residue with the non-integral charge found. So it means charge added onto the whole protein structure. So there is no problem in charge adding. Okay. So this is the first part. You have done. Congratulations. You are going very good. You have done the first part successfully. You have prepared your protein structure for docking. And now the next part is you will have to prepare the ligand as well. So in that case, don't go to the file. No, you cannot incorporate the ligand into that interface by the file. There is an option ligand over here. So click input and then open. Oh, that is important. You have downloaded the ligand into the PDB format. This Autodock, it accepts the ligand into the PDB-QT format. But you don't have PDB-QT format of your ligand. So what you will do? Oh, don't worry. We have downloaded a very good software. That is called OpenBabel. So go to the OpenBabel. It will help you. So I have downloaded the OpenBabel. It is here. In my case, I am using the MacBook, so it appear like this. In your case, it may be appear differently. So go to the Babel, and here you will give the input file. The input file option is PDB, so you can select the PDB format. Where is the PDB? This is the PDB, protein data bank. Okay, so you can, yes, by clicking the input, you can click the PDB, lig.pdb. This is the PDB format, lig.pdb. This is the PDB format. So you will click on this and open. So yes, this is the path where your ligand is present. I have already discussed what do you mean by path. In my case, that is the user. In your case, that may be the C drive, and then your documents and then Autodock, etc., 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 where your folder is present. In my case, it is user, Muzammil, desktop, docking tutorial, and in the docking tutorial, the ligand name is lig.pdb that is present. So this is the output format. So you can go here and then select the output format pdbqt. Where is the pdb? Yes, this is the pdbqt. So you can click the 3D and then you can click the convert over here. So I will prefer to use the open babel by the command line. So command line is very important. I will use the command line. You can also use the command line by the if you have a MacBook, you can go to the terminal, and if you have a window, you can go for the CMD, and then a black window will appear in your window. In my case, I'll use the terminal because I'm using the MacBook. So in this case, if you have opened the terminal, again the path is very important. You will have to give the path. How you will give the path? In you will have to cd change the directory. You will type the cd change directory, and then you will give the path to your folder at where you are working. Like copy the path, and by just typing cd, and then you will paste that path over here and then click. So it means you are in that specific folder where your ligand is present. Okay, so in I am here in the folder that is docking tutorial. I have already, yes, this is like this was the path. In my case, it was the cd, then desktop forward that is the backslash, and then docking underscore tutorial. This is my working directory. In your case, it may be different. So the important thing is how you will convert your PDB, your ligand which is present in the PDB format to a PDBQT format. So for this, you will use the open babel. In open babel, you will type the command o babel first, o babel, then space, that is minus or hyphen, then I for input. It means your input file format, the format in which you have downloaded your ligand. So we have downloaded our ligand into the PDB. So I will type -i p d and b. What I have done, I have typed the p, d and b, and then give me a minute please. Okay, this is, I have made the command for you as well. I just copy from here and paste here. If you have some problem in resolution or some problem in watching, so you can change the video quality to 720 or 1080. Then you will be able to watch, or you will be able to see all these things very clearly and easily. So open babel. After that, our input format is PDB, so we have type -I PDB, and the name of your PDB file, the name of your ligand file. In our case, that is lig.pdb. In our case, in which format we have downloaded? Guys, it is very easier. In which format you have downloaded the ligand? In the PDB format. So -I PDB, it means our input format is PDB. And then the name of the file. The name of the file is lig.pdb. In your case, it may be whatsoever the name is. It may be different, but that name will be ended .pdb. It represents the format. So the next thing is, in which format we want an output. We want output in PDB-QT format. So we will type -O, the format of the file, the format of the file in which we want our ligand, that is PDB-QT. So we will type -O PDB-QT and then space and then -O. That is important. When you will type -O and format, and then after that you will have to type minus then O and then name of your file. So let's suppose I want the output file name is new ligand, new underscore lig. So this is the file name I want. So when I'll enter this, yes, successfully one molecule is converted. Now we are going to the folder, and when we will check, yes, this is present, new underscore lig .pdb-QT. It means we have converted our ligand which was present in the PDB format to PDB-QT. We can do it from the command line and also by using the GUI interface of the OpenBabel. So after, one thing is again very important. You can minimize the ligand as well by in the OpenBabel. Yes, you can minimize it. For the minimization, you will have a different command, like this is my command for minimization. If you are not interested in the minimization, no problem at all. No problem at all. You can skip it. But if you want to minimize your structure, you can give this command as well. That is ob minimize -ff. That is the force field. The force field, they are important to minimize. If you don't know the force field, just go to the Google and type the force field for minimization. Then you will learn what is that. For the minimization of your ligand, you can do this. -ff, that is the force field. The force field which I will use to minimize the ligand, that is MMFF94. That is the name of the force field. You can amber, you can use the amber, you can use the OPLS, and other. There are many force field which are available. You can check the force field available into the Babel, and then you can type that force field in this, and then you will give the name of the ligand. What is the name of our ligand which we have prepared? Yes, that is the new was in capital. Yes, new was in capital. These things are very important. The name, correct name is very important. You will have to give the correct name. If you have to type something in capital, it should be in the capital. So PDB-QT. The name of our ligand was new underscore lig.pdb-QT, and then greater sign, and then I have typed the lig underscore mini, MINI, mini for minimization, then PDBQT. I want the minimized structure by this name. It means this is my output format. I want the output file that will, after minimization. If you have minimized your structure, after minimization, the output file name will be lig underscore mini.pdbqt. You will enter it. Yes, congratulations. You have done it successfully. You have minimized your structure of the ligand, and the name is lig underscore mini.pdbqt. So we have converted our ligand to the format which is acceptable to Autodock, that is PDBQT. So now come to Autodock again. This Autodock interface is containing our protein structure. I am just repeating it again for you. What we have done so far, we have, from the file, by the read molecule, we have selected the protein structure, and then by the edit we have deleted all the water molecule, and then we, from the MISC, we have checked the missing atom, and then we have repaired the missing atom, and then we have added the hydrogen by clicking the hydrogen, then add, and then by selecting the non-polar we have added the hydrogen, and in the end we have added the Coleman charges. Okay, so now we are going to the ligand, and then input, and then open. So it is showing this ligand name which we have just converted and minimized. The name is lig mini.pdbqt. So we will open it, and that is very important. When you will open it, it will automatically added the hydrogen and the charges which you have added to the protein structure, because you have prepared your protein structure, and these things will be applied automatically to your ligand as well. When you will click it, just look at what happens. Yes, this window shows added the gaseous charges, found 19 aromatic carbons, detected 4 rotatable bonds, and the torsion is set to 4. So just click OK. Yes, in the green, this is our ligand, the green. So we will just go to the top and compress this chain by clicking this, and the detail residue will disappear, and here we have the ligand. By clicking this, it is little bit more visible to you. Okay, so this is our ligand which we have uploaded into this GUI. Okay, so after this, again go to the ligand, click the ligand, and go to the torsion tree. By clicking the torsion tree, go to the detect root. You are going to detect the root of your ligand. This highlighted, green highlighted, is the root of your ligand. You have done what you have done. You have just clicked the ligand and torsion tree and click the detect root. So this is the root which is detected. This green, this is the root which is detected. Okay, again go to the ligand, go to the torsion tree, and choose the set the number torsion. So when we uploaded our ligand to this interface, the message was shown that our ligand contain 4 rotatable torsion angle, 4 torsion angle. So it is again showing 4. So you can dismiss it. There are number of active torsion are 4. Okay, dismiss, and then come to the ligand again, click your torsion, and choose the torsions. So we have choose the 4 torsion and the maximum capability is 32. Autodock can deal the maximum rotatable bond, that is 32. It means if you have a long ligand like the long peptide, so Autodock will not be more suitable for that docking. So because it just can it just you can say work on the 32 rotatable bonds. So after over 32 we have just 4 rotatable bonds in our structure. Okay, click on done. Okay, so now we have prepared our ligand as well as the target structure. So now we are going to the making the grid macromolecules, macromolecule and choose. Okay, so the name of our protein is alpha gluco. We will choose this alpha gluco and then click the select molecule. This window will open, just it is showing some information, contain non-bonded atom, found 3500, 36 non-polar hydrogen, etc, etc. Click OK. So now if you remember, you have uploaded the structure of your protein in PDB format. So we have prepared our structure by deleting the water molecule, by checking the missing atom or repairing the missing atom, by adding the hydrogens, polar hydrogens, and by adding the charge. We have prepared it. So now we will save this structure into the PDB-QT format, as we have converted our ligand into the PDB-QT format by the open Babel. So we will now save our prepared protein structure into the PDB format. So I am giving the name to the structure. Here you can give the name like I am again giving the name new, only new. No, it will be overlapped and you will be just puzzled. So my name is Muzammil. I am just giving it Moz, M-U-Z-Z, M-U-Z, Moz. So the new structure, that is the type PDBQT. File type will be PDBQT. The structure will be saved. The prepared protein structure will be saved into the PDBQT format. So the name of the structure is I have given the name MUZ, Muz. Okay, save. Oh yes, just you have if you have noticed, the color is changed. The color of the protein is changed. You have saved the protein into the PDBQT format by the name Moz. Yes, this is the talking tutorial folder and this is the structure moz.pdbqt, which we have saved here. Okay, so now we are going to make the grid. So again, click the grid, set atom types, choose the ligand. What is your ligand? The ligand name is ligmini. Select the ligand, click OK. Choose the ligand. Why it is showing some problem? You can choose your ligand by clicking the come to the grid. Okay, give me a minute. By clicking a grid you can go to the set atom, and then choose the ligand. Okay, so you can click the grid and then set map types, choose the ligand. This is the ligand, select this. Okay, so now we are going to the grid box. We have choose the ligand, and then we have choose the grid box. I have already set the dimensions of the grid box. This is the, okay, I'm just making it some bit clear for you. Okay, that is the protein. Yes, it is now little bit clear. Yes, so this is the ligand which is present into this cavity. So you can select the cavity by yourself. If you know where is the binding site, the binding site will be the all the portion or the site where the already bound ligand is present. Where the already bound ligand is present, that will be your binding site. So you will select the grid around that binding site. So you can change over here by changing from this. You can change the box size. You can make it small, you can make it large. It should contain your ligand. So this is, so I have selected the box around the ligand. You can find out the information of the binding site, and then you can make the grid around that site as well. Another thing is, you can pick any atom. If you know the residue which play important role in the mechanism of the protein, you can just select that residue from here by the picking atom, and then the atom will be picked, and you can pick any atom and the grid will be around that. So now in my case, it is little bit disturbed. So you can click the center of the ligand. So grid will be around the center of the ligand. So like this. And if you don't know anything about your protein, so you can make the grid that will cover the whole protein. You can make the grid box around the whole protein. That will be called blind docking. It means you don't know the exact cavity where you want to dock your ligand, so it can be docked anywhere. So that will be called blind docking. But we are not going towards the blind docking. We are just making the grid box around our ligand, because we know that. So yes, we have made it. We will just fix these things, make the little bit small. It is higher. Okay, from here we can adjust the length. Yes. So now, no, this portion of the ligand is outside of the box, so we will cover all the ligand. Yes, now it is okay. Now it is okay. This box cover the whole ligand as well. So after that, you can go to the file and then click the close the saving current. Okay, the current dimension that is saved, and then go to the grid output and save the grid parameter file, gpf, grid parameter file. Give the name to it. So I'm just giving the name to it m mean Muzammil. That the file name will be m.gpf, grid parameter file. So I am making simple for you as well. I am typing the grid.gpg is the grid parameter file. So click save. Yes, grid parameter file is saved into you can check, you can go to the folder and can check there where is the grid. That is the grid parameter file. Okay, so after this, go to the run, click run auto grid because you have prepared the grid. Click on browse. That will lead to you to your folder of directory, and click the auto grid 4 and then open. Then browse and click the grid.gph file. In the first line, you have click the browse and click on the auto grid 4 file and then open. In the second row, you have just click the browse and the file which we have now created, the name is grid.gpf. Just click on it and open it. Okay, so that will be shown here. This will be automatically over here, grid.glg, that is the grid log file. Just click on launch. So this window will appear. It is making the grid. So when it will complete its work, this will be automatically disappear. Yes, it is disappeared. So it means your grid file has been generated, your grid has been generated. Then go to the docking, macromolecule. In our case, we are going to rigid docking. Rigid docking means your protein is rigid. Your whole protein is rigid. It is not flexible. Whether we have some option, we can make some residue flexible as well. But in today's tutorial, we are working on the rigid protein docking. So set the rigid file name. In that, if you want some part of the protein flexible, it means they are free to rotate. Those residues are free to rotate. So you can make those residue flexible. Okay, but in our case, we are working on the rigid protein ligand rigid docking, rigid body docking. Okay, so I'm going to do it again. Dock, then go to the macromolecule and set the rigid file name. So the name of our protein was MOS, which we have converted or save. The protein name was mos.pdbqt. So we will open this mos. Okay, so after this, go to the ligand, choose the ligand name is lig.mini, and then here accept it, and then again go to the docking, search parameter. There are many parameters, so we are the genetic algorithm is very important. I'm not going to the detail because it is already very long tutorial, so I'm not going into the detail, but the genetic algorithm is very important for the search parameter. That will be searching the binding pose and all these things. So genetic algorithm is important. You will click on the genetic algorithm, and then this thing is very important. The number of runs, the default is 10 and the population size is 150. But if you want good results of your docking, so make it 50 and the population size is 300. It means the 300 population size and the run will be 50 run will be generated, and then you will be able to find out the best pose or the best orientation, how your ligand is oriented into the binding site and making the interactions. So but in today's tutorial we will use the default, that is 10 and 150. So click on accept and remain keep the other things like as default, and just click on the accept. Then after this, when you have you have done this, so then go to the docking and accept, and then again when you will go to the docking and then output, and in output there are many genetic algorithm. There many things can be selected, but the best is Lamarckian GA. You will select this, and this is the docking parameter file. So you can give any name to this docking parameter file. So for your ease, I am just giving the name to doc. So the file name will be doc.dpf. Like we have GPF grid parameter file, so this is the docking parameter file. So it will save by this name. Okay, we have saved it. So we have saved the docking parameter file by the name doc. So now we have generated the file, and after the generation of the what we did when we generated the parameter file for the grid, we clicked on the run and then we clicked on the auto grid. But now we have generated the docking parameter file. So again we will go to the run and then we will select run Autodock, and from here you will browse, you will click browse, and then you will select the Autodock 4 exe file which you have placed in your folder. Remember, I have told you to place two files which are very important for docking. Place them into the folder that was Autodock 4 along with the Autodock 4 grid 4. So now we will select Autodock 4 and then open and then browse. The name of the parameter file for docking was doc.dpf. Just click on this, and this is the log file name will be doc.dlg, docking log file. Just go click on launch. Dear students, you have launched your docking. Now the window will appear, and when it will disappear, it means your docking has completed. It will take some time when your docking will completed, and after this there will be some file. If you will look into the folder, go to the folder, and you can see there are some map file and other file as well. So now you can analyze the docking results. So click on analyze. You can analyze the DLG file by here as well directly. This is doc.elg file. You can open it into the text editor or WordPad. This is your docking docking log file. It will show you the results. All these things will be shown, and if like this is the docking parameter file. When you will open the docking parameter file, just find RMSD table. RMSD table. When you will find RMSD table, so you will hear. This is the RMSD table. So it shows you have these ranks, and the run 7, it shows the maximum energy, that is maximum binding affinity, that is minus 7.06. that is the energy in kilocalories per mole. And if you want the inhibition constant Ki, you can also find Ki as well. Ki inhibition, you can click the inhibition. So this is the inhibition constant Ki, 11.41 nanomolar. So in this way you can also find out the inhibition constant as well. So this was all about the docking. So when you analyze the result, just go to the edit, delete all molecule, and then continue. Okay, so when you will delete all the molecule, just go to analyze, docking, and open. So this is the DLG file. The ligand is shown over here which you have used, and then macromolecule, this. I has changed the directory, so it is showing like this. Analyze. What you I'm doing it again for you. Delete all molecules. Okay, so analyze. Just I'm going to set the directory which I have changed, because I was thinking there is some problem in the result. So let me check it. Tutorial, set the directory. Okay, so now go to the analyze, docking, open the DLG file. The ligand will be shown. What is happening? Open DLG file. No. Okay, so this is the ligand. So now again go to the analyze, open. When you will click open, automatically the protein which you have saved, that will open. So this is the complex. So now you want to analyze it, whether the conformation. So go to the conformation and play. In my case, there are some problems, so therefore it didn't dock anything. It is not showing the docking results. Let me change the directory. Set the directory. There is another file that is named this is the DLG file. This is our ligand, and then analyze, macromolecule, open. So this is macromolecule with our ligand. I'm quickly going to the conformation, play, ligand, set. So these are different conformation which is showing here. I'll show you the ligand in this format. Okay, so when you will click here, this is showing different conformation. The ligand is docked into different in the 10 conformation, because we have given 10 runs. So it is showing 10 conformation. This 1, 2, 3, 4, 5. Okay, so you just click on this. So here there is built hydrogen option, hydrogen bond. Click on this. So there is the hydrogen bond, and that is shown here. This is the hydrogen bond, and the distance of the hydrogen bond is also shown. I'm just making it easier for you to this. Okay, now look, this is the hydrogen bond, and the distance of the hydrogen bond is 2.0. that is making with the fifth conformers, and the 6 is making 2 hydrogen bond. The length of the one hydrogen bond is 2.17 and the other is 2.217. So it is showing 2 hydrogen bonds. So that is important. If you want an interaction or analyze your result, you in this way you can analyze the result. And if you click on this show info, so the conformation, yes, conformation 6, the energy is this, ligand efficiency is this, inhibition constant is 19.19 nanomolar, intermolecular energy is this, electrostatic energy, blah blah blah. There are a lot of things, lot of information is available. So that, dear student, this is the end of this tutorial. In the next tutorial, I'll show you how you will able to make the 2D diagram, or the diagram which you will be able to publish in your research paper. So that was all about the docking. If you have any question, you can ask easily into the comments or comment section. You can suggest any other topic on which I'll make some other videos. If you like the whole tutorial, please subscribe, like and comment on my channel. And one thing is very important: if you haven't worked before on the docking, so in the step where I have minimized the energy of the ligand, don't do that step, and accept that step. Other are good. You can go with that. By OpenBabel, you have to just convert the ligand into the PDBQT format, and the protein structure that is important, that is present in the PDB, you can convert it by the Autodock as well. So you can practice it. If you have some problem, you can ask your question in comments. Take care. Allah Hafiz.